The Biological Research and Services Laboratory, or BRSL, is one of the four research and technical laboratories of the Natural Sciences Research Institute in the University of the Philippines, Diliman. The BRSL began as the electron microscopy room under the supervision of the RASL. It used to house a transmission electron microscope that was donated by the Japanese government and established to provide services related to microscopic analysis of biological samples, mostly with the use of the TEM. The operation of the EM room eventually became independent from the RASL. And when the Biological Services Lab, or BSL, was formed in the early 1990s, the TEM technician, and room were absorbed by the lab. Since then, the laboratory expanded its research and services thrust such that in 1999 it was renamed the Biological Research and Services Laboratory, or BRSL. It has undertaken researches in the fields of medical genetics, molecular biology, biotechnology, environmental genotoxicity, and DNA analysis of bioarchaeological samples. It also provides biological services, mainly in the evaluation of toxicity and safety of foods and drugs. The lab has trained numerous students, teachers, researchers, and other professionals in basic molecular biology techniques, laboratory animal handling, and plant chromosome preparation through its workshops, and offers thesis support, student apprenticeship, and biological assistance to clients, especially from the College of Science and the university, as well as from the public and private sectors. In 2007, the BRSL pioneered the holding of a regular workshop on the proper care and use of laboratory animals in compliance with Philippine Animal Welfare Act and the Department of Agriculture Administrative Order Number 40, which is an essential component of the Animal Care and Use Program of UP Diliman. In 2018, the Molecular Biology Workshop was revived with modified activities and up-to-date methods, supported and funded by Office of Extension Coordination of the UP Diliman Office of the Vice Chancellor for Research and Development. In 2019, with the changing times and increase in number of researchers and staff, the BRSL conducted its strategic planning workshop, and from this point, the lab is governed by its vision of becoming a leading laboratory in biological research and technical services in the Philippines and in the world. And its mission summarized into 5 Ps: promote, produce, perform, provide, and protect. Additionally, the laboratory's first ever microalgae workshop was held in June of the same year in cooperation with the Philippine Phycological Society Incorporated, focusing on the isolation and purification of freshwater microalgae. In the same year, the Advanced Molecular Biology Workshop was also organized for the first time in September to provide progressive concepts on molecular and genetic engineering through a series of lectures and hands-on activities. In 2020, the BRSL co-sponsored the ASEAN Data Science Explorers Enablement Session together with the ASEAN Foundation and the College of Science Computational Science Research. Currently, the laboratory conducts free webinars and online consultations that encompass disciplines of biological sciences during this difficult time of pandemic. With support from the NSRI administration, the college, and the university, the BRSL also boasts of its recent awards, recognitions, media exposure, scientific papers, and conference presentations of its members. The BRSL, now composed of 23 researchers and staff, mostly project-based, continues to develop its equipment and facilities in order to improve its services and enhance its research, and fulfill the laboratory's advocacies of providing excellence in biology research, serving the Filipino community, and promoting the advancement of science both at the local and international arena.
All right. Thank you very much for that introduction of BRSL. That's who we are. And of course, a pleasant morning to everyone in cyberspace, especially to our speakers and audience. We welcome you to the eighth episode of the Biological Research and Services Laboratories webinar series with the theme Karagatan para sa Kinabukasan, Exploring the Wonders of Philippine Waters. I am Nikki, your MC for today. So before I begin, here are a few reminders we have to observe for today's webinar. Please turn off your microphone and video camera during the presentation of our speakers unless requested. Please note that the webinar is being recorded during its whole duration and is being streamed in Facebook and in YouTube. We would like to see and hear your comments and questions. The chat box is open to accommodate these, and we will be entertaining all of it during the open forum. We encourage everyone to stay and join the photo documentation by the end of the webinar. And for those who will be needing a certificate, you need to accomplish the evaluation form, whose link will be shared in the chat box by the end of the webinar as well. So to formally open today's exploration, may I call on the laboratory head of BRSL, Dr. Pierangeli G. Vital. Ma'am Pai?
Thanks, Nikki. Good morning, everyone. I formally welcome you to the eighth of the Biological Research and Services Laboratory of Natural Sciences Research Institute, University of the Philippines, Diliman webinar, or simply BRSL webinar series titled Karagatan para sa Kinabukasan, Exploring the Wonders of Philippine Waters as part of the efforts of BRSL to contribute to the understanding of science, particularly biology, and its allied fields in the community. Welcome back to those who have been with us in the last 7 episodes. For this webinar, we are fortunate to be joined by experts Dr. Lemuel Aragones from the Institute of Environmental Science and Meteorology, UP Diliman, and a colleague from the Philippine Phycological Society, Inc., Dr. J.V. Saco from Batangas State University. Maraming salamat po sa inyo for accepting our invitation. Akmang-akma ang ating tema ngayong umaga sa init ng panahon na ating nararanasan. So excited na po akong mag-swimming sa karagatan at makinig sa inyong mga lectures. I'm sure excited na din po kayong lahat. But before that, let me acknowledge the following. This webinar will not be possible without the support of our NSRI Director, Dr. Elmer Stashot, and the efforts of the amazing and hardworking researchers and staff of BRSL. So earlier, you already saw the available resources and services that our lab offers. Although our lab is still physically closed, we are accepting online consultations and continue to organize webinars such as these. So please feel free to email or contact us if you have biological concerns. Also, stay tuned with our BRSL Facebook page for updates. With this, I wish you to have a productive webinar, and with our great speakers for this morning, I am very sure you will. Thank you very much, and I turn you back over to Nikki.
All right, thank you very much, Ma'am Pai, for welcoming our audience. Tama nga naman, sa init ng panahon, sobrang akma ng topic natin for today. And we all know that more than two-thirds of the world is water. And just when we thought it couldn't get more interesting, new findings arise and suggest otherwise. From the algae that floats with the current to the marine animals that actually swim with it or against it, for sure, dagat na dagat na yung iba sa atin dyan, and hopefully, our speakers today can bring this heat to you as we dive into our topic. For the first speaker, he is an assistant professor and center head of the Verde Island Passage Center of Oceanographic Research and Aquatic Life Sciences, or VIP Corals, in the Batangas State University, where he helps in providing a world-class research, training, and extension services on the marine resources and marine environment in the Verde Island Passage. He obtained his bachelor's degree in biology from the Pamantasan ng Lungsod ng Maynila, his master's degree in the Marine Science Institute here in the University of the Philippines, Diliman, and doctorate degree in cell biology from the Graduate School of Integrated Arts and Sciences in Kochi University in Kochi City, Japan. He has been involved in different scientific engagements, locally and internationally. Not to mention, he received different awards, including the Balik Scientist Award by the DOST Picard in 2020 and the DOSTP Card Publication Incentives Award last May 3, 2021. To give a whirl on the wonders of seaweeds, making your ew to an A, let us all welcome Dr. JV A. Sacco.
Thank you very much, Nikki. Gusto ko yung from ew to A to ow. All right, so can you see my screen now, guys?
Yes, sir. Very clear.
Okay, thank you. Hi, Doc Pai. Thank you very much for the invite. Okay, so let's just make this very informal. So parang this way of encouraging, of course, students and other faculty and researchers to venture to marine science. My title is Seaweeds. Ano yun? Lumot sa dagat. Ew. Most of you for sure yun yung naiisip nyo pag naririnig nyo yung seaweeds or lumot sa dagat, ika nga nila sa Tagalog. No? And I think this webinar is very good because it will be a contrast of a more charismatic species as marine mammals and not so charismatic species as seaweeds. No? So, okay, so let me start by showing you a picture of me way back in, I think, during my master's day in UPMSI. So as you can see here, this is one of our site in Bolinao, Pangasinan. It's it is a Sargasso bed. As you can see, I'm lying down because medyo pagod na during fieldwork. And as you can see, hindi naman siya ew, pero siguro yung iba na medyo hindi familiar with this, yun yung maiisip nila. So let's go on with the journey on how can we understand seaweed better. And siguro at the end of this talk, siguro mas ma-amaze kayo with the wonders of what seaweed can bring us. Okay? So these are the three commonly seaweed species being eaten by humans. So andito yung Caulerpa, which is lato, gusto, which is Capaphycus. Then we have this Codium. So pwede ba tayo maging interactive among the audience from this Zoom? Sino na yung kumakain sa inyo as part ng food nyo or diet nyo ng seaweeds? Can we do that to make it a bit interactive? So I just have to have like idea kung sino yung may idea about the seaweeds as food for their diet. So okay, so we have two participants raised hands. So pwede ko silang i-acknowledge, then I could ask them. So can I acknowledge si Feresa?
Hello.
Hi, Feresa. So ano yung common mo kinakain dito among this picture?
So taga Cebu po ako. So sa
Okay.
lato at locally it's known as lato, yung Caulerpa.
Okay, yes. So I mean everyday mo siyang kinakain or time to time mo lang siya kinakain?
Pag malapit lang sa dagat.
Okay, yes, definitely. Pero I mean like for example here in Luzon and even kahit sa palengke dito sa Manila, may meron silang binibentang lato. Even nung nag-ikot kami sa QC before pandemic, so yun, makakakita naman ng lato. So I think that's one of the common seaweed species na binibenta sa palengke. Thank you, Ferreira. Thank you for answering. All right. So let's go to what are seaweed. So I am pleased to show you this IAC material that we developed when I stayed in Japan. So I'm one of the translator of this IAC material. This is one product from a aquarium agency in Okinawa, Japan. So basically we'll talk about what are seaweeds. So let's go now. What are seaweeds? Okay. So seaweeds are technically a marine macrobenthic algae. Sorry, narinig niyo na ako? Okay. So basically, they are exclusively marine species, naked to the eye, and they are usually attached to a substrate. And ang very fascinating, kung makikita niyo yung picture, ang daming different species at different yung itsura nila, and they are all seaweeds, no? So from a 1 centimeter size of a seaweed into a 35 cm. And actually, meron pa tayong counterpart na mas malaki sa temperate region. Like, it's a forest, like a kelp. So meron din naman tayo like sargassum beds naman ang tawag sa. So aside from that, they are photoauxotrophic organism. We're in photo, meaning they use the light on converting water and carbon dioxide into oxygen and carbohydrates on the process of photosynthesis. And one more unique feature of seaweeds is they also acquire nutrients from surrounding water through diffusion. Okay, so kaya siya tinawag na photoauxotrophic organism. All right. So another unique feature of seaweeds is they are ancestor of terrestrial plants. So as you could see here on my slide, on your left is a seaweed and on your right is a plant, higher plant. So as you could see, parang almost meron silang the same structure, no? So they have similar structure na leaf, ang tawag sa seaweed ay blade. Then of course sa higher plant it's leaf. Very unique sa seaweed because that blade is photosynthesizing as well as with leaf, pero yun nga, they also absorb nutrients and gases. No? So they also have stipe or stem-like as counterparts sa terrestrial plant, which is a stem. So ang difference nila is sa higher plant, nag-transport sila ng water and nutrients, while the stipe in the stem-like structure in seaweeds, they support the blade. They also photosynthesize if they have pigment, and they also absorb nutrients and gases from surrounding water. Then also they have hold-fast or root-like structure. So sa higher plants, mainly for absorption lang siya ng nutrients and gases. No? But in seaweeds, they called hold-fast, and it's for more of an attachment lang siya. But if there's a pigment, they could still photosynthesize and they could still also absorb nutrients and gases from surrounding water. So, but the main big difference between the two plant is seaweeds don't have fruits and flowers, no? So, kung walang fruits and flowers ang seaweed, paano sila nagre-reproduce, no? So, they reproduce asexually and sexually. So, asexually is through their spores. So, pag nag-release sila ng spores, even yung smallest spores na yon, kahit hindi siya nag-mate, tutubo siya as a one individual. And another unique feature of the reproduction of seaweed is yung fragmentation niya. Once maputo lang siya, that piece, yung nakat na dun sa main seaweeds na plant, pwede siyang tumubo into another individual. So that's one more unique feature of seaweeds. And they could also produce through sexual reproduction. So pag meet ng female and male gametes, then it will produce a zygote. Alright, so saan po natin makikita ang seaweeds? Basic requirement for seaweed to grow is, of course, dapat may light. As long as there are light, pwedeng tumubo ang seaweeds. And of course, there should be a substrate for them to attach. Pero may mga seaweeds kasi na floating. So as long as there are light. Okay, so sa seaweed, they are usually divided or practically divided into 3 major divisions based from their pigment. Okay, so we have green seaweeds. Kanilang dominant pigment is chlorophyll A and B. So tingnan nyo, very diverse yung kanyang morphology. So from flat to filamentous to calcareous and to coarsely branched. Okay. Then we have this brown seaweeds. Ang kanilang dominant pigment is xanthophyll. So as well as from flat to leathery type of seaweeds. Then we have red naman. So red seaweed, ang predominant pigment nila is phycoerythrin. So they have the dominant pigment is phycoerythrin. Pero mapapansin nyo with this pictures, bakit yung iba is color green? Bakit color brown yung iba? So ang nangyayari during their photosynthesis, yung kanilang depending on depth kung saan sila tumutubo, usually yung pinaka ginagamit nilang pigment during photosynthesis is possible chlorophyll A, kaya siya nagiging color green. Pero ang predominant pigment pa rin niya is phycoerythrine. So di ba very fascinating siya. It could change its color, but still same species pa rin siya. Alright, so in the Philippines, according to Trono 1990, we have 820 seaweed species. At during 2015 by Ang et al., nadagdaga siya to 166 seaweed species. And recently, there's a publication of Lastimoso and Santianes in 2020 that we have at present time 1,065 seaweed species. So hindi ba very fascinating yun? Napakadami palang seaweed species in the Philippines pero parang tayo nine-neglect natin siya kasi parang ew, parang it crawls on my legs pag pupunta ka sa dagat. So medyo talagang may iwi feeling siya. Pero kung makikita mo, this 1,065 seaweed species, among these, ano yung pwede natin ma-develop for possible na drug, for cancer, or for COVID, pwede rin sa COVID kasi may antiviral property ang seaweeds. Alright, so syempre matanong nyo, bakit bigla nag-increase yung number ng seaweed species? Syempre, of course, technology changes and mas kaya na ng technology natin ngayon to identify identify seaweed species na mukhang magkamukha pero different species pala siya. Alright. Ano namang ecological importance ng seaweed? So they are source of food to higher organisms. So as you can see here, itong mga catfish na to, they are eating on a hydroclathrus and panday na. Ano pa yung kanilang ecological importance? They are shelter and nourishing ground. So as usually kasi pag sa intertidal area, this seaweed species is intermixed with seagrass. So as you can see here, may mga seagrass species dito. So seagrass and seaweed beds are usually habits for other marine organisms for their shelter and nursing ground. Another unique feature of seaweed is they stabilize the substrate. For example, this cauler pa na ito, so yung kanilang root system is parang fibrous yan, parang buhok-buhok. So binabind niyang mabuti yung substrate para hindi siya mag-erode. And another good feature of seaweed, like if you've been to Boracay, so mapapansin nyo very fine yung sand, pinong-pino, and of course, it is very famous because of that white sand. And for sure, iba sa inyo, ngayon na ito maririnig, but that fine sand of Boracay came from a calcified seaweed species, itong Halimeda species. So, di ba? Something new sa pandinig nyo siguro. And of course, ito, siguro isa din sa hindi nyo pa ito naririnig. Merong isang seaweed species, itong Crustose, at least I think it is a group of seaweed species, no? Crustose coralline algae. So itong mga Crustose coralline algae is very important because kung wala itong Crustose coralline algae, yung larvae, yung coral larvae, hindi siya magsisettle sa reef for them to grow as a coral. So kung walang Crustose coralline algae, hindi magsisettle yung coral larvae, so hindi magkakaroon ng coral reef, no? So very important seaweed, specifically itong Crustose coralline algae. And of course, they are also a good carbon dioxide sink. Okay, sige, pakita tayo ng picture ha. So ano dito sa pictures na ito ang gumagamit ng seaweed? So let's try to make it interactive ng kaunti ulit. So can someone raise their hand and could someone pinpoint which among in the picture uses seaweeds? Sige na, let's be more interactive. So sino po among the audience dito po sa Zoom? Kahit isa lang po, taas ng kamay, then sabihin among which among the things in the picture uses seaweeds. Wala. Okay, si Jericho. Sige, Jericho from BRSL. Okay.
Hi sir,
Hi.
nabasa ko before yung ice cream ay gumagamit din ng seaweed daw. Totoo po ba yun?
Yes, totoong-totoo yun. Hindi yun daw. Totoong-totoo. So tama si Jericho. Thank you for answering my question. Sige, yes, tama. Seaweeds, ang ice cream ay gumagamit ng seaweed. Pero lahat ba itong nasa picture ay gumagamit ng seaweed? Yes or no? Yes, all of them uses seaweed and even a fabric Yung curtain na nasa likod uses seaweed, even yung fire extinguisher ay gumagamit ng seaweeds. And yes, Jericho, tama ka, ice cream uses seaweed. So explore natin later kung paano. Alright. So from the ecological perspective, ngayon naman we go to the economic importance of seaweeds. No? So yes, basically they are being used as food. So kung mahilig tayo sa Japanese food, you are eating seaweeds, which is specifically nori and undarya, which is yung nasa ating miso soup. So sa Philippines, ang basic natin na kinakain is itong lato and guso, which is yung nandito sa right side of the picture. So aside from that, it is being used as food supplement kasi pwede siyang, kung gusto mo magpapayat or pwede mo mag-increase ng weight, you use seaweeds as a good food supplement. So aside from that, it can be used like for example, pickled seaweed. So these are Filipino product, itong pickled seaweed and shingaling seaweeds. So itong big sheet naman is parang snack naman siya. Dito, galing ito sa Thailand. And pwede rin siyang gamitin as a marmalade. Alright. So bukod sa atin, bukod sa tao, nakaka-benefit din ang ating mga animals. So they can be used as animal feeds and fertilizer. So in the Philippines, actually, before ang Philippines is top producer actually ng euchrematoid species, which is Capaphycus and Euchrema. This is a red seaweed kung saan we are extracting them because of their karaginan. No? So ipakita ko sa inyo kung bakit napaka-importante ng karaginan. So these are sample picture of a seaweed farm in the Philippines. I think this is in Zamboanga. But the nearest one here in Luzon is in Batangas, which is in Calatagan, and in Palawan. No? So, bakit isipin nyo, tanong nyo bakit tayo hindi na number one? Because we are being overtaken by Indonesia because possibly yung culture technique natin medyo outdated na because seaweed farming began in 1970. And some of our cultivars or yung seedling na itinatanim natin sa farm ay strain pa nung 1970, so siguro hindi na siya ganun makapag-adapt sa ating changing environment. So pero bakit napakalaga ng karaginan na ine-extract natin dito sa red seaweed, which is Capaphycus and Euchrema? So karaginan are being used as stabilizer, emulsifier, thickening, gelling agent. Okay. So makikita nyo dito, these are food and they are mostly manufactured in bigger scale. So, pag walang karaginan, for example, pag walang karaginan ng milk, pag ininom mo siya, mararamdaman mo buo-buo because nagka-curd siya. But because with the karaginan, tinatanggal niya yung pagiging curd para maging smooth yung feeling mo. So, kung iba sa inyo, mahilig uminom ng beer or juice, pag wala rin karaginan, magiging hindi smooth yung feeling. So, for the example din naman ng ice cream, i-compare nyo ang ice cream with dirty ice cream and commercially available ice cream. So makikita nyo with the dirty ice cream, medyo may crystal yung formation niya. As compared with commercial ice cream, very smooth yung kanyang texture. So that's why, because meron siyang karaginan. So totoo si Jericho na may karaginan po. May seaweed po ang ice cream. So aside from that, oops, sorry. Okay, aside from that, they are being used as pharmaceutical, no? So, as you may know that our Muslim brother and sisters, of course, hindi sila pwedeng mag-take ng animal-derived na product. Kasi yung mga capsule available in the market, usually they're animal-derived product, no? So that's why since this is plant-based, dahil seaweed nga siya, so they was able to develop a capsule from karaginan. So aside from that, they are being used in industrial application, no? So sa mga sanitary napkin natin, disposable, air freshener, toothpaste, healthcare products, and even batteries because ang karaginan is good conductor of electricity. So aside from karaginan, so meron pa tayong iba pang extract na very economically important. Like for example, itong alginates naman. So ito yung sinasabi ko sa inyo na can grow as much as 25 feet. So ito yung mga kelp sa temperate region. Ang counterpart niya sa Philippines is itong sargassum, pero medyo hindi siya abot ng mga 25 feet. Medyo mababa lang siya. So anong use nitong alginates na ito? So syempre, number one, ginagamit siya as beauty products. Can be used as ink for printing. Pwede rin siyang wound dressing, dental, impression molds, and for casting. No? So ano yung pwede nating ma-research about sa seaweed? So of course, basically, identification of a certain species. Kahit sabihin mo na meron ng 1,065 seaweed species, pero that is just only a number and hindi mo pa siya actual na nakikita sa field. So pwede ka mag-identify ng seaweed species sa certain area. So for example, bibigyan ka ng picture, ano ito? So you identify them based from their external morphology and cross-section ka for the internal morphology. Then you use the aid of the microscope to identify them. And of course, you could able to publish that identification. So, anong ginagawa natin pag nag-identify tayo ng species, no? So, syempre, pag nasa field tayo, this picture is in Verde Island, no? So, from our recently concluded externally funded from an NGO in Japan. So, for example, ito, kukuha ka ng sample, syempre you have to sort them according to species for herbarium voucher. Then after that, siguro during sorting, no, pinipicture mo na rin siya para meron kang documentation. Then after that, you mount them and press them for herbarium voucher. So bakit natin kailangan i-deposit sa herbarium? Pero because this will be our evidence na ito yung na-collect nating samples or ito yung na-collect nating seaweed species. On your left picture, ako yan, nagsusort ako ng mga different seaweed species, and this is the herbarium in UPMSI, which is called G.P. Velasquez Phycological Herbarium in honor of the father of phycology in the Philippines, Gregorio T. Velasquez. No? And ang pioneer nitong herbarium na ito is si Dr. Chono, which is our national scientist. At ang nag-digitize nitong herbarium na to is aking former supervisor na si Dr. Edna Fortes. No? And on your picture sa left, on the right picture, so ito yung mga actual specimen na namin sa Batangas State University. So I will try to develop also as well a physical facility of herbarium in Batangas State University. Alright, so anong pwede natin sagutin sa area? To identify seaweed species in a specific area, to have information on what species can be found in the area or during what season. Accounts, actually, ang herbarium ay magandang accounts of past geological and environmental phenomena kasi there's a certain seaweed species that could be a good bioindicator of a certain global phenomenon. No? So, as well as a conservation initiative for future generations, specifically yung mga endemic seaweed species sa Philippines na wala na at present time, like for example, yung Gracilaria manilensis. Isipin nyo, way back 1970s, maraming seaweed species sa Manila Bay. And they was able to find endemic species in Manila Bay, kaya siya tinawag na Gracilaria manilensis. Unfortunately, ngayon, wala na siya as my recollection. No? So, with such facility as herbarium, doon nakadeposit yung ating only proof na merong Gracilaria manilensis. Okay? Enable others to study or verify existing information. So aside from that, ano pang pwede natin magawa? So community assessment. What are ito more of more of how much the seaweed species cover the area. Okay? So this one is community assessment. What are the species in the area? So we are using line transect quadrant method. This is very basic, pero syempre ang part na pwedeng employ dito is yung ma-train yung eyes mo on identifying seaweed species and how to assess them based on their cover. Kung gaano kalaki or gaano kalawak yung kino-cover ng seaweed species sa area. Okay. So it could encompass like intertidal area, itong mababaw, and even subtidal area. So pwede ka mag-scuba diving. So, for example, itong aming recently concluded na project from Nagao Environmental Foundation of Japan. So, we able to assess seaweed and seagrass community in 4 strategic area in Verde Island. No? So, just a quick information. Sa Verde Island, ipinangalan yung Verde Island Passage, which is the World Center of Marine Shorefish Biodiversity. Bakit siya pinangalan sa Verde Island? Because itong islang ito yung nasa gitna mismo ng passage. Okay, so very strategic itong area ito. So we was able to identify that Padaina is thriving in all areas in the island. So ano yung pwede natin masabi with Padaina species? Actually, ito Padaina species is a bioindicator of increased nutrient in the water. And possible na mag-reflect ito sa isla because walang magandang sewage system sa island. And most of our site is malapit sa coastal barangay with may domesticated domestic animals such as yung pig pen. So yung mga waste nila is dumidiretso sa dagat. So one indicator ito na possible na mataas ang nutrient ng tubig using Padaina species. Okay, so ano pwede nating masagot dito? So able to monitor species, specifically economically important. Thus could scheme a management plan on harvesting wild stock if culturing is not possible. No? So able to monitor changes in the community composition through time as affected by different physical chemical parameters. Like for example, yung pinakita kong Padaina species, maganda siya i-monitor through time. Like for example, a 5-year or 3-year plan, kung mag-change siya within the area, at least meron tayong ma-reflect natin na ano yung current state ng lugar. Then able to monitor the effect of damage to communities such as oil spill and coal spill. So ano pang pwede natin mapag-aralan? So aside from identifying seaweed species and the extent of the cover of seaweed species in the area, we could study them through their photosynthetic activity. No? So we could identify the effect of salinity, temperature, pH, and light. So we could use the picture on the upper right. These are oxygen evolution technique, and this is the basic and traditional way of measuring photosynthesis in seaweed, yet very effective. No, and on the lower picture, this is a lower picture. This is pulse amplitude modulation fluorometry, and this is the most updated and modern way of measuring the photosynthesis in seaweed. But with my opinion, both these two methods should be used during your experimentation. Okay. So, ano yung pwede natin mapag-aralan? So, first is meron akong papakita sa inyo na the study namin, which we are very fortunate that it was awarded third best student poster from PPSI Symposium. So we was able to assess yung effect ng thallus morphology sa photosynthesis. As you can see here, merong group A na mas mataas ang photosynthesis. Ito yung mga maninipis na seaweeds. And itong mga group E, medyo mas makakapal itong mga calcified. So makita nyo mababa yung kanilang photosynthesis. So basically, this just showed that photosynthesis is more affected by their thallus morphology. Okay, so pasok naman tayo doon sa internal structure ng seaweeds. No? So makikita nyo dito yung different position ng seaweeds, different chloroplast position ng seaweeds. So makikita nyo rito, yung isa is nandito sa isang area lang, then sa kabila is different yung position niya. And we was found out that these two seaweed species, magkaiba yung kanilang photosynthetic activity. At ang maganda dito sa dalawang species na ito, we observe that lagi sila magkasama sa natural habitat nila. So siguro kaya magkaiba rin yung kanilang photosynthetic activities para hindi sila mag-compete with the resources within their natural habitat. So fortunate tayo that this was able to awarded best published paper by Japanese Phycological Society, no? And another is ito is recently published namin to na work on the chloroplast movement in seaweed. So isipin nyo ba, nagmo-move pala ang chloroplast sa seaweed? Yes, nagmo-move po sila based on their light condition. So pag daytime period, so yung seaweed, yung chloroplast is nasa surface while during nighttime is nasa side wall. And makikita nyo dito sa graph na ito, magkaiba rin ang kanilang photosynthetic activity. So maganda itong input if we're going to culture or if we're going to cultivate this seaweed species because it will be practical na i-conserve mo lang yung light mo during daytime period at hindi mo na siya extend until night. Kasi may mga seaweed species kasi na longer period sila na nagpa-photosynthesize. Alright. So, fortunate po tayo that this was awarded Best Oral Presenter in recently concluded National Symposium ng PPSI, and nadala po natin to representing Philippines in the International Civil Symposium. So, anong pwede nating masagot dito sa ganitong method for our research? So, able to identify species if sun or adapted, sun or shade adapted, able to identify the tolerance of species to specific physicochemical parameters. This is very important because with this, we could able to determine yung optimum condition for culture and cultivation nila. So aside from that, syempre pwede tayo mag-culture. So itong nasa left picture is ito yung actual seaweed laboratory in UPMSI wherein I am connected with the National R&D in seaweed before way back 2014. No? So ito yung kinukulture na namin na seaweed species. And on your right picture, ito yung mismo ang aking kinulture na monostroma in Japan. So as you can see, I was able to document their life cycle and I was able to have a full use seaweed na pwede na siyang culture at pwede siyang siguro dito on pwede siyang i-outplant for cultivation sa open water. Alright, so ano pwede natin mapag-aralan with that culture? So siyempre pwede natin mapag-aralan yung reproductive phenology nila. For example, kasi sa seaweeds kasi specifically for red seaweed, medyo maganda ang ganda ng life cycle nila kasi meron silang tinatawag na tetrasporophyte. So makikita nyo, 4 yung spores niya in one spore. Then we have carposporophyte. This is parasitic form of reproductive structure in the female seaweed species. And of course, spermatia, which is yung sa lalaking gametes. And we have the vegetative, which is hindi pa siya reproductively mature. Then, anong implication niyan? Siyempre, implication niyan is for us to develop a culture technology. So, meron tayong pinakita dito na we was able to train before personnel on possible culture technology in green seaweeds. No? Yeah. So, itong publication na ito is a good input. We was able to characterize the photosynthesis and growth of Monostroma latissimum, and it is a good input if we're going to improve the current farming technique of Monostroma latissimum. No? Okay, with that, marami pong salamat, and if you have further question, I'm open for Q&A, and I have my email here if meron kayo gustong itanong further, if gusto niyo about sa method na ginagawa namin sa lab, so you could email me directly. So that's jv.ako@g.panstate.u.edu.ph, and you could visit VIP Coral's FB page and website. So yun po, marami pong salamat.
Alright, thank you very much, Doc JB. Napaka-interesting po ng topic nyo, no? And sobrang diverse. Talaga namang naging A ang dating iyo.
Oh, thank you. I love that.
Okay, so for those who already have questions or comments for Doc JB, please just drop it in the chat box with your name and affiliation. We'll be raising all of it during the open forum.
Ay, okay, sige. Thank you.
And thank you, Sir JB. Okay, for our second speaker, he is a professional and current director of the Institute of Environmental Science and Meteorology here in the University of the Philippines, Diliman. He received his PhD in Tropical Environmental Studies in Marine Ecology at James Cook University in Australia, and he is also the head of the Marine Mammal Research and Stranding Laboratory of UPIESM, working on the marine biology, ecological sustainable development and conservation, and management of marine mammals and other large marine vertebrates. Of course, here to present about the lessons what we can get from the marine mammals in the Philippines. Let us put our hands together for Dr. Lemuel V. Aragones. Doc Lem. You can see the screen now.
Okay, I hope you could see my slides now.
Yes, very clear, Doc.
Hear me? Okay, maraming salamat for the invitation to be one of your speakers for your 8th webinar series. umaga na ito ay gusto ko sanang bigyan kayo ng init ng dagat galing sa malayong lugar sa ating marine offshore areas, karamihan ng ating marine mammals natatagpuan. At ang ilan ay sa tabing dalampasigan din, tulad ng mga ilan na pag-uusapan ko later. No? So ito po ay isang making lecture tungkol sa or my take on what we have already learned sa mga lessons that we have, the science that we have developed from looking at strandings ng marine mammals sa Pilipinas. And of course, I would like to initially acknowledge this is a work of labor of love and labor of networking. And I've been fortunate to have been molded by quite a few professors in my master's, bachelor's, and PhD, and been fortunate to work with quite a huge array of scientists from biologists, marine scientists, environmental scientists, vet, medical doctors, physicists, and the like. So it is imperative that I I mention all of this and acknowledge them right from the very start. Okay, so having said that, let's now proceed. So my talk will be, I'll just give a brief introduction on what marine mammals of the Philippines are and the Philippine Marine Mammal Stranding Network. I'll showcase some of the data that and how many of these have been turned over into research opportunities in terms of publication, production of mentoring of graduate students, and even undergraduate students, and focus on stranding data per se, and then the other sets of data. And this was the conclusion is what I work on most of the time last night. Is it's something that I have never done before. So I'm trying to summarize what we have done so far from strandings. So let me start by telling you na may ilan tayo, there are about, say, 90 cetaceans in the Philippines, and we have 1/3 of that. And there are quite a few of the seals and sea lions or the pinnipeds, pero wala tayo nun. Meron tayo niyan, nasa Ocean Adventure. Meron tayong tinatawag din na mga sirenians, 4 na klase po yan sa buong mundo, pero may isa lang po tayo, yung dugong, nakikita sa Indo-Pacific. Parang bangus lang yan. May bangus sa India, bangus din sa Pilipinas. So, and of course, there's quite a few set of aquatic mammals, but they're not truly aquatic in the sense of it. But I also often include that in the introductory context just to enhance, hopefully, interest on this small clawless otter in particularly found in Palawan. So here in the photo, you can see a spinner dolphin, a melon-headed whale, spotted dolphin. Let me just enhance my pointer. This is a rhesus dolphin, of course, a dugong, a spinner dolphin, melon-headed again, and spotted. And unfortunately, these marine mammals strand. And let me clarify, marine mammals generally are charismatic, but as an environmental scientist as well as a marine mammal scientist, I would like to think of this group of animals to be players or components of a very complex set of ecosystems. Some of them are partly involved in seagrass, coral reef ecosystems. Some of them are offshore towards the pelagic, neritic part of the offshore ecosystems or oceanic ecosystems. So here are some of the variety of marine mammals. On the lower right is a dugong, which is often found on coastal nearshore areas because of their affinity to seagrasses. And this is one of the largest marine mammals in the Philippines, toothed marine mammals in the Philippines. This is often referred to as a sperm whale, or for exaggeration, the giant sperm whale. And here is a rare, without the stranding in effort, we would not have discovered that we have a common dolphin. And here is the a Fraser's dolphin, an example of a a unique case of taking care of young calf of, and this is a rhesus dolphin. And rhesus dolphin often usually have in adult form, they're linear scars, and you can obviously see that this is a calf and it doesn't have yet those scars. So they strand for variety of reasons due to natural causes as well as human-induced causes, of which I will tackle one by one as the talk goes on. You know? Just to introduce you to some of the terms, a stranding is when marine mammals are run aground or they are often caught in shallow waters and they cannot go back. Hindi na sila makalangoy dun sa mas malalim na lugar na kung saan yung kanilang tunay na habitat. And that is what a stranding. A stranding is often taken as coming from the Old English word called strand, which is practically another word for beach. No? So kaya yung strand sa mga English, British, Australian, it's just another word for the beach. Sa atin sa Pilipinas, no, na we have abused the word stranding. Sabi natin, oh, na mulan lang dito sa Quezon City ng isang oras mahigit na malakas. Stranded na tayo sa mga lugar natin, hindi tayo makali sa baha. No? And therefore, there is this confusion of how to use the word stranding. Anyway, since sila ay marine mammals, hindi po sila isda, sila ay humihinga, one of the reasons why scientists believe these animals prefer to strand themselves when they are weak or sick is that they need to breathe. Yun ang difference ng marine mammal sa mga isda. No? So you can often see a stranding to be alive or dead when they are beached already on the shore. They can be isahan like this dead bald-nosed dolphin, or this is a live, of course, false killer whale. And then there is the single or mass. This is a recent mortality of a mass stranding of Fraser's dolphin just this year in Del Gallego, Camarines Sur. And there could be out of habitat like this, what we had in 2010 in Manila Bay, or unusual mortality events wherein there's a spread of mortalities of dead animals, often even live animals, within a geographic area. And we have already had this in certain areas like Lingayen Gulf. So having said that, I must give me a few minutes to or seconds to explain the network. Came back after my PhD from James Cook University, trained to do aerial surveys, ship surveys, tag, and all those things, but didn't have access to that. So I was bewildered and so nangangapa paano ako magiging pagkadalubwa sa aking napiling group of animals to study. So fortunately, I was able to be part of a group of passionate people who develop or establish the Philippine Marine Mammal Stranding Network, which is a consortium of mandated agencies, local government units, the industry that created the technology, ocean adventure, and the academe UP, Diliman, where I belong. Okay, so that is the network of the Philippine Marine Mammal Stranding Network. And let me just build the big picture that we have regional chapters in all chapters that have all regions that have coastal administrative regions or administrative territories. Okay, so now the story is that we PMMSN started in 2005. We will have our anniversary this coming October, and believe it, now we have a data set of at least 1,176 stranding events. These are stranding events. The number of animals are almost twice the number of that. And then you could see the annual increase in the first 4 years. It was an average of 30, and the next 5 years was an average of 60. And since 2014, we have an annual average of a little over 100 per year. So we have more or less stabilized in the 100 annual average since 2014. And from this data set or database, we've been able to now understand better the species composition of the marine mammals that we have in the Philippines. Mind you, it's very hard to study, survey, and do all those high financial capital outlay, manpower that is necessary to do all the surveys of a very wide million square kilometers of exclusive economic zone of the Philippines. So through this stranding database, we've been fortunate to see that the spinner dolphin is the most common strander, and based on the Peterson context of the equation of mark capture recapture, we could almost say that this species is the most common and most abundant marine mammal in the Philippines. Is closely followed by the Fraser's dolphin. These two are deep or offshore species. They prefer very deep habitats. And the dugong, mind you, is a Sirenian. However, it is alarmingly um, important to say this, that given that they are another critically endangered marine mammal in the Philippines together with the Irrawaddy dolphin, they are far more getting more alarming rates of stranding per year. And the cetaceans, I would say, would be in the thousands to even hundreds of thousands of in terms of population. Maybe these are my best guess, scientific guesses. But dugong may be not in that sturdy pop numbers, maybe less than, if not in the thousands at the most. So the dugong is getting a bit of an impact and without this, we would not be able to say that. And so the other species are obviously as follows. And you could see at the end of those, we could see that there are very rare stranders, such as the killer whale. As I said, without the stranding database, we would not have been able to see that we have a common dolphin, a ginkgo-toothed beak whale, a Direyanigala beak whale, fin whale. These are very rare and almost some of the least known, except of obviously for the killer whale, one of the most known in terms of research elsewhere. Okay? So given that, pwede pa lang ang dami nating makukuhang information sa stranding. Initially pa lang, yun pa lang, mahalaga na yun. Ano? And so in terms of information that you want to inform mandated agencies, the accessibility of this scientific information, we also look at this in the regional context. And you could see the ones with the asterisks are the regions that are far more having more frequency, having more frequent cases of strandings on an annual basis. This is led by the top notch is Region 1, next is Region 5, and those are in Luzon alone. And the third is in the Visayas, Region 7, okay, Central Visayas. And the rest is all, there's no exception. All regions in the Philippines, even the Bangsamoro Autonomous Region or mostly Mindanao, have, and even the National Capital Region. And you could see that the color of the bars is based on the seasonality of December, January, February, March, April, May, and so on. So you could see, and you could imagine now that the March, April, May has, if you total all of this, seems to be in the pattern is that they have the most strandings, and also it's the proportion of live strandings seems to follow such pattern, and that the more there's strandings in certain seasons, the more there will be live strandings. In all practicality, ang ating live stranders ay about 60%. Some ranges about 50 to 60%. But that is unusually high in comparison to the rest of the world. And that's why there is so much interest in looking at the strandings, particularly in the Philippines, of which we have somehow become a model to the rest of the world, especially in our region, wherein we were able to establish as well. We've been the pioneer to push for the establishment of the Asian Marine Mammal Stranding Network. And using this stranding data set, we've been able to, again, using ArcGIS, spatial tools in looking at trends and patterns, space and time, we were also able to use or use a tool in the artist called fishnet, and we played around with the pixel size, and we thought we settled at 15 kilometers by 15 kilometers, which is more or less where we want to inform, because that's more or less in many cases the coastal area extent of um, municipalities or cities. And you could see that in throughout the Philippines, there's so much, and the pixel is 1 to 4. First green, the yellow have 5 to 8 stranding since 2005 to 2020. And imagine that 15 kilometer by 15, you can see that there seems to be some high preference or preponderance of occurrence in certain areas, and that's why we call this city or municipal hotspots. And I chose the top 5. Number 1 is Santa Ana. Santa Ana is in the right smack tip of northeast Luzon, followed by Badoc, which is the same area actually. The top 8, I think, is Claveria, close to Badoc, as well as Santa Ana. And then you have the Gupan, of course, the Gupan area here in Lingayen Gulf. Then you have Pagudpod again up in the northern tip of Luzon. Sorry, this is where Badoc is, Badoc Ilocos Norte. And then you have Alaminos, and these are all within Region 1 and Region 2. And those are some of vital information that we have been able to generate, particularly in the interest of protection and management. And as a network, as an annual national network, we have deemed it important to share techniques, protocols, development of reports, informing mandated agencies through the years. And now I would like to look at the other data sets that we use in the context of using these strandings as research opportunities. And one is we evoke the One Health paradigm. And this is very relevant sa panahon ngayon na pandemya, no? Now we see the importance of One Health. Siguro in the near future, sana magkaroon tayo ng One Health Commission in the Philippines as a starter to be able to, yung mga medical doctor nakikipag-usap sa vet, yung mga yan, nakikipag-usap sa environmental scientists, nakikipag-usap sa social scientists, etc., etc., to be able to capture that the context of the idea that the health of people is really, truly connected with the health of the animals and our shared environment. So that's the One Health paradigm. Of course, we include here our marine mammals, particularly the dolphins and whales, which are referred to as using these marine mammals as sentinels of ocean health. What is the concept of sentinels of ocean health? Kasi itong mga dolphins, whales are long-lived. Some of them can live beyond the whales. Some of the whales can be as long as greater than 70. Some of the dolphins can live that long. The toothed dolphins, some of them 30 years. So still relatively long. And therefore, they have that capacity to reflect what they have assimilated in their habitats. And see, this illustration captures it all, the effects of persistent contaminants, the debris, eutrophication, the increasing radiation, the increasing sea level, ship traffic, the noise, the effects of this, all of this in the marine mammal populations. Species can decline or become functionally extinct, some can persist, and all of these are further mediated by algal blooms, harvest overfishing and bycatch, fisheries depletion, introduction of invasive species, and epizootics. These are all now relevant in the context of the pandemic that we see. Okay? And therefore, again, what we have been finding out, seeing in the results of our studies through strandings, we can now almost reflect what their environments are in, depending on the species. So let me start with the microbiological findings. I've been fortunate to been able to mentor quite a few PhDs and masters and even undergraduate students. So one of my very first is in the Philippines is Kinabukasan, who been very prolific as a publisher, in terms of my former PhD mentor mantra, publish or perish. So this is just a simple table showing the context of the things that we have done as in collaboration with other scientists. And you see that Windel Rivera, Ma'am Oxy Siringan, etc., have been part of the regional PhD advisory committee. Dr. Masangkay of the College of Veterinary Medicine have all been helpful in all of this. And so we could been working on Leptospira, toxoplasmosis as well, and other bacteria. And in some cases, we have all of the samples, we have been able to see and detect bacteria and the like. And therefore, these are interesting stories that would not have been possible without access to the research opportunities given by these standard marine mammals. And obviously, I must acknowledge that we are barely scraping the surface. Based on the total number of strandings, we are still not that efficient in collecting all the samples. It's the logistical challenges is a nightmare. Alam niyo naman yan, yung ibang region, ang lalayo ng central regional offices nila where the team originates most of the time. But we've been trying our best to expand the network's capacity in localizing it. But still, it's a work in progress. One of the things that I truly enjoy as a scientist is finding answers and finding interesting cases and always tickle my fancy as a scientist. Oh, wow, nung nakita ko to nung una namin ginagawa, ang laki ng implication ito on taking care of rehabilitating stranded marine mammals, because now we've seen even the third, fourth generation bacteria, some of the animals have already, bacteria found in these animals have already have resistance to those bacteria. These bacteria have already resistance to antibiotics, implying the very long journey of these antibiotics from the land base to the deep offshore, such as these are all offshore species. So yan ang sinasabi ko sa reflection of the sentinel species as sentinels of ocean health, these dolphins and whales. Sir, so this is just a break on all the monotony of the graphs, just to show you, bring you back to the beautiful creatures. These are some of the exceptional cases. Tala, the rough-toothed, has been an exceptional case. We took care of her for 50 days before she was released. Very successful story of rehabilitation. This is a sad story of one of the most critically endangered cetacean in the Philippines, the Irrawaddy dolphin, a victim of bycatch. And here is another interesting case, a very young calf, neonate baby dugong called Bughao in an area very close to my heart in Busuanga, near Calawi. Next to that, we were able to, and I got another PhD, Jonah Bondoc, who's part of the NSRI on another lab. And we were able to look at, develop answers to some of the unique stranded dolphins, such as a beak whale and a kojid. These are offshore species again, and we were able to look at the histopathological context and explain some of the possible reasons of why strand. And on the way, we have recently been accepted. Our paper this is a technique paper, method paper. In a PhD, you really have to tweak all the techniques to fit on to, since blubber is so involved in our study, and that the machine and then the pollutants is this PCBs that we were looking at is a fat-loving thing. We have to tweak the procedure and develop a technique for validation and application of a cleanup method. Okay, so that will be coming out soon. And this is one of the papers we're working on right now. This is a product of our PhD showcasing just the simple message here is yung mga PCBs, okay, hindi ma-unload ng males kasi hindi sila nagpapabatutoy, hindi sila nagpapadede. Okay? So yung mga females, yung mga adult females, they have lower concentration of these toxins. And that is a very important, and that is very, very much relevant to our scenario here. And that is also validated by other studies overseas, especially in highly urbanized and polluted bodies of waters in Europe as well as the Atlantic. Another work that I've been fortunate to be able to work with are the vets. Marami akong vet friends, and some of the best vets in marine mammal medicine is involved in the PMMSN. And I've been fortunate to work with Leo Suarez, develop blood values for spinner dolphin. One, as I said, siya yung pinaka-common nating strander, pero sa encyclopedia ng mga vets sa marine mammal medicine, wala yung reference values yung blood niya. So we were able to develop that from rehabilitated spinner dolphins. And again, I've been fortunate to be affiliate of the College of Veterinary Medicine and be part of a committee to guide the master student, Dr. Sandilin Shu, a Malaysian. She's back home in Malaysia now, but she did a great contribution to marine mammal medicine. And we looked at a clinical pathologic assessment of select stranded marine mammals in the Philippines. It's a great, and we're working on a few publications as well. And of course, I was fortunate also to be working with applied physics. We did the master's on environmental science in our program. Annelien Lagi, I call her HL for short. And we were able to look at some environmental correlates to see association with the patterns of strandings. And we use the 6 or the 5 geographic big biogeographic regions is the South China Sea or the West Philippine Sea, the Sulu Sea, the Celebes Sea, the Southern Philippine Sea, and Northern Philippine Sea, and then the internal seas or the Visayan Sea or Visayas region. And the internal seas involve the Visayas Sea, Camotes Sea, Tanjung Strait, Bohol Sea, etc. And then these are and you can see the pattern is that most of these strandings in the internal are quite unique in the internal waters, very close to the South China Sea. But in terms of, this is in terms of sea surface temperature, but in terms of chlorophyll, which can be a proxy for their prey, you'll notice that most of the time the strandings are in with very low chlorophyll because obviously the chlorophyll has already been transformed into other trophic levels for biomass wherein these were eventually probably consumed by the dolphins wherein they are associated with certain biogeographic regions. So these are some of the findings that, and here I would like to showcase 3 species that looked when we looked at abundance and distribution sort of questions. This is a striped dolphin, this is a spinner dolphin, and this is spotted dolphin. So here on the left is a map showing all the different stranding plots of the plotted in different colors. Okay, green is for the spotted dolphin, the red is for the spinner dolphin, and yellow is for the striped dolphin. And these are all stenellids, meaning ang genus nila is Stenella. So collectively, they're referred to as stenellids, magkakamag-anak sila. And you can see that there are clearly important clusters, which now we are writing a paper, developing this to identify important marine mammal areas. And since these are some of the things that we believe can also we could infer from strandings to inform conservation. And we also believe that this is a great way to project the distribution of the stenellids nationwide. But these are important marine mammal or cetacean or stenellid areas. Similarly, I selected the dugong, also very close to my heart because it's the animal I've been working with since time immemorial. And this was where I started my marine mammal research, and this is the um, an area of concern because this seems to be now almost isolated from this group, which is probably a larger chunk of population, still hopefully continuously exchanging. There's still a gene flow in that, as well as probably this is probably exchanging genes with the population in Indonesia. So this is a bit of quite of a challenge. Remember, there is a split here of the current of the from the equatorial current, creating the very strong Kuroshio current. So they're very, very critical that conservation efforts concerning dugongs take from sort of clue or key cue from this initial findings. And we are publishing something towards that. So as I said, these are the animals that I looked at, and with my RA, HL, and a colleague, Dr. Armanapan, to do some GIS, fancy GIS things. And of course, the other one was the dugong. This is one of my favorite dugongs, Aban, a regular dugong, being accessed by some of the eco tourists in Kalawit. So I would like to challenge myself of all the time as a scientist, and with so much data that I'm looking at, I see the opportunity to ask questions, especially those that are very rare. And this in the left are some of the very deep diving marine mammals, such as the sperm whales in the color green, the pygmy sperm whale, color yellow, the dwarf sperm whale, or brown, the blainvilles, beak whale, cuviers, ginkgo, longmans. These are all very rare and yet seems to be stranding all over the Philippines. And we're trying to understand that. But it appears that the nature of the archipelagic nature of the Philippines with very deep drop off, very deep average depth of bodies of waters, except the areas here in Visayas, as well as the connection here probably in the Cuyo group of islands connecting to the Panay, are some of the shallower, as well as the Bicol shelf areas here. But the rest are very, very much of the neritic area already, 200 meters. And this graph is on the mass stranding and near mass strandings. And you see that these are, again, information that we could not have generated without the stranding effort. So in short, we have learned so much from strandings. So with that, let me conclude. So um, this is how I would like my audience today to bear as an important message. This is the first time for me to, as well, last night, to synthesize what we have done so far. Um, in terms of um, in terms of the Philippine marine mammals, we know that there are groups of stations in dugong in the Philippines as early as the first publication that I was involved is 1990 by Steve Leatherwood, who actually was one of the key persons that inspired me to pursue this field further. Um, and then through strandings per se, we further the understanding of these marine mammals. We now have understanding of annual trends of strandings. The species has been expanded because of having now listings of more variety of species through time. We are seeing important spatial patterns regionally, biogeographically. We may develop the biogeographic regions as we look at the population genetics, seasonal patterns, strandings, live bed, and name it. These are quite a few of things that we are actually already doing and available for analysis or further analysis. Another thing that I discussed briefly today also is about the context, capturing the One Health paradigms in relation to the Sentinel context. So we've been able to look at the microbiology of the organisms or microbiological context of these marine mammals, the ecotox. I have now PhD student Kim Benjamin who's looking at endocrine disruptors for the female reproduction. We do a lot of histopath and we have a set of vet med students, undergrad, that I will co-advising with this semester. And we have still building on the mythological reference values of certain species that we have. Uh, Dr. Leo has so much to work on. There's so much accumulation already of data. The work of Doc Sandy in clinical pathology, the pathophysiology interest of Dr. Cristorno. We will all pursue that. We are looking at so many opportunities to identify quite the interesting parasites in the ears. Lately, a very compacted intestine of a very recent strander of false killer whale in the region 1, and so on. There's a recent paper that we will come out with characterizing the a simulation of open water dynamite blast, simulating dynamite fishing and its potential impacts on marine mammals. We are also of interest on the marine debris that these animals. Cetacean morbillivirus in the advent of COVID-19. We should start monitoring this, and we have already developed a proposal, and we're been asked to develop a full proposal now. Dr. Kinabukasan will also be the point person on that. And therefore, the sentinel species have truly been able to highlight that our offshore marine environment is already compromised. And then we also look at the conservation in terms of, now we can picture distribution, some important key information on relative abundance, which are more abundant than others. As I said, we are trying to develop a publication in important marine mammal areas. We would be looking at environmental and oceanographic correlates to strandings, feeding ecology with collected otoliths, beaks of all those collected in the intestine or stomachs of these marine mammals. We have now a few students who I will be co-supervising with some of the geneticists at the Institute of Biology who would look at population genetics. We have a lot of samples for natural history, aging, and looking at reproductive success, anatomy of the ears, physiology of the things that we see during the rehab process. We can now have some idea of their conservation status based on threats. And most importantly, this information are accessible immediately to the mandated agencies who truly need them. So I hope I've been able to capture more or less the ones I discuss are with Astrid. Maraming salamat po.
Okay, thank you very much, Sir Lem, for that interesting topic. No, I think ang dami pala nating pwedeng makuha from mammals that are just simply stranded, unfortunately stranded, that is our seashores. And so we are now formally opening the open forum for our questions and comments. And might I say that ang dami nating tanong. So everybody's hooked up with our topic for today, both for Sir Lem and Sir JV. So may I now invite for our first question for a while. She doesn't want to be introduced, but this question is for Sir JV. Sir JV, how are seaweeds cultivated by seaweeds farmers that are being used for commercialization? How long will it take for them to harvest?
Okay, so in Philippines, yung pinaka kinocultivate natin na seaweed species is Capaphycus inducuma. This is widely cultivated since 1970. Kaya siya ganun ka effective na organism or seaweed species because mabilis siyang tumubo. Yung original concept ng seaweed farming is ganito. Noong 1970, yan, nagkwento na ako. Sorry, Sir Lema, kwento lang ako ng kaunti. Noong 1970, tumaas yung demand for this seaweed species because of their carrageenan content in extract. So, nangyari sa Philippines, yung mga tao nag-harvest sa wild. So, anong nangyari? Na-deplete yung stock from the wild. So, that's why na-formulate or na-formulate yung pagkakaroon ng seaweed farming. Yung seaweed farming is actually parang na siya, monoline, tali lang siya. Tapos ilalagay mo lang siya sa dagat, tapos itatay mo lang yung maliit na branch ng galing doon sa seaweeds, no? So tutubo lang siya within 30 to 45 days, yun ang first cycle nila. So continuous yun. So pwede ka mag-produce ng maraming tons of dried seaweeds. Then after yung raw material, i-dry mo siya. Tapos that raw material usually binibili siya ng ating international partner kasi sila na nagpo-process sa kanilang lugar. Pero meron naman tayong processing plant dito sa Philippines. Meron kasing Philippine natural grade of Karagatan. So that's why very popular siya because mabilis ang turnover ng product and madali lang siyang gawin. Unfortunately, with the seedlings that we are using now, very susceptible siya sa mga disease like yung epiphyte, yung dumidikit na mga ibang seaweed species or blue-green algae dun sa talus ng halaman. And yung ice disease. Yung ice disease yung parang pamumuti ng talus. So ngayon, marami naman ang ginagawa, specifically ang DOST kasi part ako ng seaweed R&D back then. I am senior research assistant ni Dr. Edna Fortes. So we was able to produce a seedlings from a wild stock. So meaning hypothetically they are genetically viable because galing ito sa wild stock. So meaning nagkaroon na ng genetic recombination from a spores from a female and male gametes, no. So meron hypothetically they are very viable. So yun na-produce namin yun. Unfortunately, I was go to Japan for my PhD. So during that time na nandun ako, alam ko hindi na-download na yung technology sa farmers. So ngayon they are still trying to make a cultivars na kayang i-resist yung changing environment natin ngayon. So yun.
Thank you very much, Sir CV. Actually, we have a few more questions for you. Sunod natin from Sir, for a while. Ayan, from Sir Jude Francisco, also from BRSL. Hi, Sir JV. Super ganda ng talk ninyo. I have a question
Thank
po.
you.
Are all seaweeds edible for human consumption? If not, how can we identify these edible species? Thank you very much po.
Okay, so technically, all of them are edible. Pero based yan sa kultura ng coastal area. In the Philippines kasi, medyo parang hindi ganun yung acceptance sa seaweed as food or staple food. Like for example, in the north part of the Philippines, in Cagayan, Ilocos pala, mababa yung kanilang cancer rate because they are taking sargassum, which is a brown seaweed, medyo matigas-tigas na seaweed siya, as part of their sinigang. So ginagamit nila yon as replacement for gulay sa sinigang, yung sargassum. So yung sargassum na rin, yun kasi, may fucoidan kasi siya. And that fucoidan is very good property to combat cancer. So kaya mababa yung cancer rate sa Ilocos. So in terms of sa buong Philippines, usually naman kinakain siya, usually they are all palatable naman, pero depending na siya actually sa kultura ng lugar, kung matagal na nilang kinakain yun or what. Kasi sometimes kasi merong, pag ang source ng seaweeds mo for food consumption is medyo questionable. Like for example, medyo malapit sa resort, so possible na mataas yung nutrient na dinidischarge from the sewage ng resort. So medyo magdadalong isip ka kasi, for example, meron kasing, not to scare everyone may namatay kasi from Bulinaw, Pangasinan na kumain ng isang seaweed, ang tawag doon ay kulot, which is Acanthophora specifera. Later on, they was found out na hindi kulot hindi seaweed species yung cause ng death, but a blue-green algae, which is Limbia ata. Limbia. I'm not sure if tama yung identification ko. So yun. So, of course, we have to know kung saan galing yung source, kung malinis yung tubig, and so on and so forth. Because, as you know, seaweed acquired their nutrients and minerals from the surrounding environment through diffusion. So kung ano yung nandoon, so pwede nilang ma-acquire. So you have to be very careful.
So malaking bearing pa rin po pala talaga yung environment na pinagkukunan ng algae for it to be considered edible po. Tama po ba?
Yes, correct.
Okay, thank you. Ayun, more on follow-up question po ito from the first question. What are the critical factors to consider in doing seaweed cultivations po?
Okay. So critical factor dyan, syempre yung light, because kung walang light, hindi magpo-photosynthesize ang seaweed at hindi siya mag-grow. Then another one is temperature and salinity. That three factor yung talaga major playing factor for seaweed to grow. So pag masyado na mataas yung temperatura, pwedeng mamatay. Pag masyado naman diluted yung salinity ng tubig, mamamatay din sila. So with Capaficus and Lucuma culture, meron silang optimum range na nakuha temperature. Like yung temperature is 29 to 31 degrees and salinity of 30 to 31 ppt. So kung ang seaweed farm mo ay malapit sa river, most likely it will not succeed because idudulot niya lang yung salinity ng tubig. So yung light naman, medyo moderate light level lang siguro around 300 to 400 micromole photometers per second, so around that.
Okay. Thank you po. These ones po are for Sir Lem naman. Sir Lem, how do you know that there are strandings in a certain place? Do you coordinate with LGUs or the residents? Follow up po doon. How do you take care of your samples from the field going to the lab? Sir Lem.
Sorry, Anoli. Sorry.
Sige.
Hindi naman tubig. Anong first question? Sorry.
Ang first question niya po is, how do you know that there are certain strandings in a certain place po?
Note that we already are about 16-year-old national network. By virtue of memorandum of agreement with BFAR, which has mandate over dolphins. And of course, my political boundaries, lahat ng strandings, we also include in the network, the LGUs. We've been very, very fortunate to have a very strong and engaged and empowered stranding network, which actually reports immediately to us. To me, I'm the hotline. I have the hotline, so I cannot replace my cell phone number since I came back. And the effectivity or efficiency has now been increased far more by the internet capacity, the cell phones, the technology of the cell phones. Now people can take pictures and the like. Ang galing na ng kahapon, isang araw nga lang, I was contemplating with my colleagues sa PMSN, say, ang tindi ng mga nagagawa ng mga stranding network chapters natin, because one chapter in an area of remote area in Masbate gave supportive care to an animal before it was released. So these are things that we taught them. We have established protocols, so it's evidence-based decision making based on our protocols, and then before they release, so they have certain parameters. So to answer it briefly, maraming paraan, and now that the Facebook is also, we have an open Facebook as a group, may close and open group kami that can report, but obviously I get the report even before the regional directors, or even sometimes I talk to the mayors and the like.
So parang malaki din po yung nagiging role ng mga chapters ng network natin in coordinating with the LGUs and reaching out din po sa inyo, tama po?
Definitely.
Yes.
We have about 4,500 strong certified responders, and about close to 150 vets that have also been certified to do medical management. There's no course in veterinary medicine for marine mammal medicine, so we have to do extensive training to harness their skills. But the first response is very, very critical. Hindi biro yun kasi some people have had the misconception na akala nila pag huminga kasi yung dolphin, nasa ibabaw yung ilong o whale, nagpo-produce na parang vapor. Inhale mo sila, then they exhale. Then akala nila, iniisip nila, para sigurong radiator to, nag-overheat, nilalagyan nila ng tubig. Hindi nila na-realize na actually nilulunod nila lalo, no? Because kailangan niyang huminga doon sa ilong. And therefore, these are some mistakes that are still being committed by those that didn't go through the training.
So maybe it's something that we can consider to be developed later on. Kasi sabi niyo sensitive training po ito eh. And for it to be more efficient, baka pwede siyang later on
May online training kami
implement.
which is through mandated agencies then. We offer
That's
that now
great.
since the advent of no face-to-face. Last year we have had virtual conferences and virtual trainings, and we still continue. Second, hundreds ang aking audience, ang aming audience, and that is allowed. You have the capacity, fortunately, because UP has a registered license room.
Yeah.
Second
That's
question.
the second question po. Actually, the second question is already answered. Paano po natin na-reach yung mga LGU?
Hindi, I think yung
For
paano
that.
pinapreserve yung samples.
Yeah.
That,
Oh, yeah, that one. How do you take care of your samples po from the field going to the lab?
ang sampling depende yan sa code ng animals. Ano? Of course, yung code 1 is still alive. So what you could get is only blood or skin that are flaking off, or take swabs. And therefore, depending on the technique that you got, the media will only work for about 48 hours. So most of the time, there was still the capacity for public transportation. Papaeroplano yan, o ipapabus, and all of that. Coordinated yan, yung mga RAs namin, kukolektahin sa bus, sa bus terminal. Most of the samples, microbiological, are done that way. Fortunately, the others can be preserved in formalin or either alcohol, that which have longer storage time before. And therefore, yun yung mga binubus, binabarko, binakargo, and then pagdating dito, gagawin ng mga RAs namin, papalitan yung buffered formalin for longer storage.
So that was during the time po, before pa po magka-pandemic, when everything is accepted.
Dati na yan hanggang ngayon. No?
Hanggang ngayon din naman po. Kahit pandemic po.
Hanggang ngayon, hindi kami makakuha ng microbiological unless yung sila mismong papagawa doon sa area nila, because kailangan ipadala dito since hindi nga tayo makapag-lab eh.
That's true. That's true po. But hopefully in the future, we can do that again. Ayan. Another question po ulit po for you, Sir Lem. Are there specific anthropogenic activities that may cause strandings of the marine mammals?
Yeah, I evaded the details kanina sa talk kasi I know somebody will ask that. Obviously, ang alam natin na mga anthropogenic activities, obviously there are things like such pollution, which we see from our ecotox studies, as well as in the context of pathogen pollution. We also have, we could see the context of infer from some behavioral context, as well as we have preserved ear bones to look at potential contribution of dynamite blasts implicating acoustic trauma to these marine mammals, especially the shallower species such as the dugong, which seems to probably the reason why they strand very frequently in the context of the collection of about 30 plus species. It's the third animal. So most likely marami ang anthropogenic context, the drivers of bakla. These are the fish corals in the shallow areas. If they're placed in areas where animals are also seems to be feeding, then therefore the animals can be trapped during the low tide, resulting even to mortality, so death or drowning of these animals. So one of the major issues namin sa anthropogenic probably is more of the human interaction. Could be on the bycatch, fishing effort has obviously increased relative to say 50 years ago, and therefore these increasing live stranders probably implicate something to that effect. But we are still trying to tease out and answering this carefully.
Okay. So thank you very much, Sir JV. Sorry, Sir Lem. We have more questions for you. But right now we have a question for Sir JV. Sir JV, this is from Christian Joseph Ong. Hello po, Sir JV. I have a question. What could be the possible studies for the molecular ecology of microalgae in the oceans?
All right. So with the current scenario that we have, di ba, very eminent na talaga yung climate change, no? And actually, they are consider marine macrophytes, specifically seaweeds, seagrass, and mangrove as winners of changing environment. So maganda natin siyang pag-aralan kung ano yung genes na could able to withstand the adverse effect of climate change. So magiging signature siya for us to study. Later on, magkaroon tayo ng idea na these are the genes that could able to survive with this changing environment. And syempre, of course, nandun yung connectivity studies, hindi naman mawawala yan. And that's very important because doon natin makikita if, like for example, an economically important seaweed species, if there are connectivity on the specific place, and with that kind of data that we could have, pwedeng for ano siya, for management ng area, specifically if that seaweed species is not yet developed for cultivation, kung wild harvesting. So maganda natin gawin din yun, no? Pero for the meantime na hindi pa kaya mag-molecular, we could study their biological aspect and when is the time, kung kailan sila madami, then kung kailan kaunti. So doon natin mabibase kung ano yung pwede nating maibigay na polisiya or intervention or recommendation sa LGU if they're going to venture to this harvesting of seaweed. Like for example, we have before during my RA time in Sorsogon, we was able to document or map the extent of Gilligella acerosa. Gilligella acerosa is a red seaweeds na maganda siyang source for agar. So yun, we was able to identify kung kailan sila madami and kung kailan sila kaunti. And during that time kasi nag-harvest na sila doon ng Gilligella acerosa because there is a Chinese investors or businessman that kumukuha ng mga raw materials. So yun, was able to establish that. So yung ganun. For molecular, marami naman pwedeng mapag-aralan dyan. But basically, it's more gearing to ano sana for cultivation because in the Philippines, number one aquaculture seaweed production. So molecular studies gearing to ano cultivation kung pwede natin ma-combine ang isang spores to another spores for a good cultivar na mas resistant siya sa ice disease, epiphytes, and so on and so forth. But actually, that's already been done. Ginagawa na siya for the meantime. Pero syempre, optimization pa ng methods or ng cultivar. So, siguro kung may mag-venture din from that molecular aspect, edi mas mabilis yung pag-identify natin kung anong cultivars yung good for being resilient sa panahon natin.
Okay, maybe si Sir Christian makakatulong sa atin dyan sa
So you could contact Dr. Michael Roleda of UPMSI because they are the one who's doing that at current time.
Okay. Thank you so much, Sir JV, for answering that question. Another one po from Ms. Feresa Cabrera. Hi, Sir JV. Any chance we're starting to explore the biodiversity of our seaweeds in the mesophotic or the 30 meters to 150 meters plus marine environments?
Actually, may mga effort na on doing that. Specifically si Dr. Wilfred Santianes of UPMSI and also collaborate din sa dalawa naming DOST Picard project to date, no? So they at least they are starting it kasi for sure marami pang seaweed species dyan sa area na yan kasi as long as there are light that is penetrating, mabubuhay sila. And very interesting kasi yung mga nakikita ni Fred doon sa nila recent cruise and field nila sa West Philippine Sea. So share niya yung mga pictures, yung mga seaweed species, and very interesting. And I think and I hope that we could also do that in Verde Island Passage, no. Pero for now kasi with our approved DOST funded project, it's more of the intertidal to subtidal area kasi yung capacity rin namin to assess the area medyo limited, and we are bounded kasi to to do survey in 5 provinces of VIP with 2 sites. So that's 5 times 2, so minimum of 10 sites to assess seaweed seagrass biodiversity. So most of them are intertidal and subtidal lang. So pwede namin i-try later on siguro. And it's very interesting kasi most of them are red seaweeds, and maganda siyang source for if you're going to do cultivation technology for this. Mataas kasi yung value ng mga reds for pigment, and ginagamit siya for cosmetic and other industrial application.
Okay. Thank you so much, Sir JV. Ito naman susunod na tanong is your a question actually for Doc Lem. So Doc Lem, may I know your take on the whale hunting in Japan?
Talaga si JB, controversial pa yung piniling tanong. Ano?
Sorry, sir, kasi actually, I'm so interested kasi, of course, yung nag-aral tayo sa Japan, hindi naman natin siya matanong sa mga sensei natin doon. So, syempre, that's of part of culture. So, I want to have like a point of view of a whale or dolphin cetacean expert on how you deal with it. No? Sorry.
Okay lang. The culture context have huge implication on as a mediating factor of the what environment we will have. Okay, so in having said that, so it's a human nature to establish that culture. It's a product of tradition. So you could see that there are certain people that will eat that prefer to eat certain species, and logically the drive fishing or the dolphin drives in Japan has been happening for thousands of years. I've actually been took part as an observer in one of the part of getting quota for dolphin catch with the scientists way back 2007, if I remember right, or early 2005. And so they're so honest. They stick to the quota. But having said that, they also have been seeing that there's a decline in the numbers. So obviously there's the impact on the population. There are always going to be ethical questions on this sort of traditions. And I'm sure quite a few of you have seen the documentary of the sea piracy. The thing is that every people will have a variety of ideas. But unfortunately, some of them may not now be backed up by scientific data or by science, simply as stated as that. Bakit ko sinabi yan? Kasi obviously, siguro nung unang ginagawa nila yung marami pa. And obviously, these species are long-lived. Mahaba, meron silang pagdadalaginding stage, binatilyo stage. Hindi sila yung manganganak agad. So, having said that, they are obviously vulnerable to overexploitation. So, ang culture also will have to change slowly, but it will not change like that. So, I know I have quite a few colleagues in Japan cetology, even younger than me, have very dynamic ideas, and they don't want to. Their federal government are actually, pinupondohan pa yung mga whaling. So it's part of the system. Nire-reduce yung price ng karne ng whale para mabili ng mga And so there is this support from the federal government. So slowly that will have to change, and the younger generation are far more now greater in proportion in Japan. But obviously, I hate to say this, but I hate to beg off the tone of this. There is also a big chunk of group of people out in Faroese Island, not Asians, that are also doing something similar, and that is also killing pilot whales annually on a drive, and so tama rin ba yun? And those are some of the ethical questions that we have to ask. These are things that the debate on the animal welfare and animal rights come to play. But I guess you are what you are. You eat you are if you eat this and you are that, and if you don't eat it and therefore you can sleep probably well. And but I have a huge range of friends from the different ages of scientists and in Japan. And the thing is, in Japan, next door you have an ocean area. Next door there's a restaurant for dolphin meat, and then that's the variety of it. And there's so many sensei who are brilliant people working on marine mammal science.
Very complicated siya, Sir Lem, no?
Yeah, definitely.
And
But
actually, to be
but
honest
it
din, nakatikim na ako
depends.
ng nung meat eh. Kasi sinabihan ako ng sensei ko, oh, you have to taste it. Okay, sensei. Hindi mo na matanggihan, di ba? But it's very complicated, so yeah. Thank you, Sir Lem.
Ayan. Thank you so much, Sir Lev. Actually, ang laki nga din talaga ng role ng culture and ng ethics with regards to this. But we just have to take it with a grain of salt. Sabi nga nila. Ayan. So another question for Sir Lem. This one is from Jericho. Hi, Sir Lem. Thank you for the very informative presentation. How does the presence of shipping lanes correlates with the marine mammal stranding? Also, what are the steps being done by our government on promoting dolphin-safe tuna practices to our tuna fishing industry?
Um, Interesting. That's a very nice question. But the shipping lanes in my lifetime, I've seen the shipping lanes in the Visayas change, and that I have been a witness of. I'm uh, half of my life been working on Tanit Strait, the narrow body of water between Negros and Cebu. And so I used to travel, wala pang aeroplano noon, to travel to those areas, and then nag-increase, nag-improve papunta Cebu to Dumaguete ng fast craft. And the marine mammals, the dolphins and whales that I see along the way have also been lost, or I haven't noticed them lately. And that's the influence of the underwater sounds created by especially this fast craft. And so that has to be integrated. I have a master student who would look at the baselines of sounds, acoustics. Hopefully, will be able to. But there's so many areas to that want their answers, their questions to be answered. So we'll see with regards to shipping lanes in the Visayas, wherein there's quite a huge population of cetaceans as well. With regards to the dolphin safe tuna, hey, this is interesting. I'm pro bono um, technical advisor of the BFAR on bycatch, and being having said that I am also advising them on how to comply with the U.S. Marine Mammal Protection Act. They are the United States fisheries products would require that the stamp of safety on their products that they buy, and that's called the list of foreign fisheries. It's a complicated thing that it's online, everything, but the questions that have to be answered are very technical and require far more research. So in short, there is no true dolphin safe uh, tuna. Most of this will be based on companies paying certain companies to give them the branding, who doesn't really have the data to showcase. Because even if there's just one gear that kills the dolphin and the process, then therefore it's not a safe tuna product. And that is the reality of it. But there is this clear laws based on the U.S. Marine Mammal Protection Act that should be the attempt should be towards reduce mortality, almost zero mortality, and that's challenging. And they are trying towards. I've been trying to do that when I was there in the United States, Toto, for a while, and that's clearly where they want to be. And so our data has to be comparable to that. And so the government is taking steps, and hopefully it will be in time for us to comply. I think it's in 2023 when in the final say if you still do not comply with all this and the data is not comparable, we will not allow your fisheries product to enter the United States.
Okay. Okay.
So it's a complex thing.
Yeah. Yeah, as you can see, yes, yes. Okay, another question. I think we have room for last four questions, but if you have more, just keep chiming in the chat box. This one is from, where is it? Oh yeah, this is from Christian Joseph Ong again, for Doc Lem. How about other pollutants, toxicants in the marine life aside from PCBs? Uh, can we consider this as a study on how healthy the marine life in the ocean is?
Um, as I said earlier, there are quite a few attempts to look at the assessment of mangrove, seagrass, coral reefs, and yet they are uh, limited. And therefore, it is even far more limited to look at the marine offshore ecosystem. And so the notion of how we use sentinel species as an indicative of the ocean health is the very paradigm that we use to imply that based on what we are seeing, um, almost all the dolphins that we looked at have has considerable concentration of PCBs. There is this alarming and also land-based pathogens such as Toxoplasma, Leptospira. These are all of land-based origin, terrestrial origin, and even the antibiotics. These are all, you add it up there. That's the level, and these are reflected in deep diving species. So it's an intriguing, and I'm really based on those data. I'm really not hesitant to say that our our offshore offshore areas have already been compromised. I have another student looking at endocrine disruptors or pharmaceutical pollutants from the pharmaceutical industries. And so that is in the context of looking at reproductive success or how the endocrines obviously are disrupting the reproductive success of females.
Okay, thank you. Another question for you, Benta ni Sir Lem. And this is from Sir Jude. The threat of infectious diseases, particularly the emerging ones originating at the human-animal environment interface, has caught health systems off guard. So his question is, what are the preparations the Philippines is doing right now, such as policy frameworks on One Health approach to combat this threat?
Obviously, I'm not part of any government DOH or and BAI in the context of veterinary. Um, I'm a professor of, of environmental science and marine mammal science in UP Diliman. And however, what I stated earlier was the notion that it's probably due time that we create an in One Health Commission to encourage the exchange of information and just develop a flow of this open discussions with environmental scientists, which is a diverse field with veterinarians, with human doctors, and make sense of how to anticipate the next pandemics, because this will not definitely be the last. And what I said earlier was we have a proposal on the cetacean morbillivirus. Um, it is a also a very infectious disease. And similarly, ganun din yung aerosol yan, yung plema rin. Kasi siyempre, mihinga sila, the others, katabi nila, malalaning sa ilong nila. And similarly, the same lang din, no, pag nag-ano tayo ng laway, kaya nakamasta, ano. And these are happening in their environment. And that has been documented in many areas as well. Unfortunately, we haven't had the capacity yet to look at morbillivirus, because the window for detecting morbillivirus is a very short window, I think of just 24 hours or 30 hours at the most to push it. So, and we have do not have such a capacity because of obvious reasons that we are focusing on something else. But I hope that that will be something that the government will obviously pursue in the very near future. And even I hope UP will initiate that, if not the DOH or the like, to just take advantage of the capacity or the experts of UP in terms of human medicine, veterinary medicine, and environmental science.
Mhm. That's true. I agree with that. Especially sa naging discussion din ni Sir JB kayo. There are uh, good purpose or health-related good purpose yung seaweed. So maybe that's something that we could work on. Maybe not as in UP, but in the government level. Kasi it's really a promising point of view to look into. Ayan. So, second to the last question, also for you, Doc Lem. Good morning, Doc Lem. Uh, Thank you po for giving a depth background regarding strandings. Is it true that the noise pollutions in our oceans is causing the increase of strandings? Is it somehow messing with the echolocation of the marine mammals? Thank you po.
Yes, I would say it is increasing. It's the obvious for obvious reasons. There are more boats as the population grow. We will have more ships. We'll have more boats out in the ocean, and they will create very different sounds and that were originally not there. So obviously that's something that is causing the problem. You see, the reason why sound underwater sound is critical to dolphins in general and whales and even dugongs is that they have a more sensitive hearing than sight. All
Mhm.
terrestrial mammals have a high acuity of vision because we are living in an air as a medium. These marine mammals, they are also mammals, we are also mammals, however, are living in a very different medium, water. And water sound travels faster, and therefore that's the adaptation why they utilize that, especially when they go deep dive in dark places where sight is senseless. And therefore they make use of their hearing instead, and therefore that is the reason why sound is often now implicated as a potential reason of stranding. It's simply messing up or masking the sounds that they are used to in terms of communicating as well as using it for navigation or in terms of finding their prey as well as their partners or their cohorts. And so this is part of very vital to their lives. And it's just like saying, wag kang mabubulag ka. Ang hirap mabuhay ng walang assistance pag mabubulag ka as an terrestrial mammal. Similarly, ganun din yung mangyayari sa mabubungol o mabibingi na mga dolphins and whales and dugongs wherein their hearing threshold will be now offset or the sounds are being masked because of a different impacts of sounds throughout their environments. We are still just starting to work on this. Our first paper will be coming out soon. So
And looking forward to that po. And so for our last question, and this question is for the both of you. What could be our what could be your takeaway or takeaway message for our audience? For those who would like to pursue or who would like to know more about your fields. Let's start with Doc JB.
Okay. So siguro yung take home ko lang siguro for the audience is ironically we're an ecologic country pero pero napakaunti ng marine biologists na nagbe-venture on the different marine organisms. So I hope na with this kind of platform of this kind of webinar na ngayon ay we are forced na nasa bahay. So siguro these are the best time na para for them to realize na we could do much, we could do more for our environment by engaging ourselves as part of it, like being on the field, being or studying different marine organisms, taking graduate school, and pursuing different training, seminar study on marine environment. No, so and currently kasi with our specialization sa psychology, we are very bumababa talaga, nagde-decrease talaga yung population namin on specializing this kind of field. So kaya ako advocacy ko talaga na to present this kind of format para mas marami ma-encourage na estudyante, na researcher na mag-venture to this kind of field. So, and of course, syempre for the dolphin and all any aspect or marine organism, ganun din, no? And of course, we are very open for mentoring, for collaboration. But I mean, lalo na ngayon, it's very easy to contact us. Diba? So, yun. And I hope may na-inspire isa o dalawa na magve-venture with this kind of specialization in this kind of field, no? And I think yun nga, although hindi charismatic ang seaweed species, pero as I've shown you, napakadami niyang pwedeng gawin at napakadami niyang halaga, both economically and ecological aspects. So with that po, ako po ay nagpapasalamat po kay Doc Pai for inviting me, and thank you very much to the staff of Doc Pai, kay Ma'am Jessa, kay Nikki, and the rest. And thank you, Sir Lem. I was able to be on the same day with you. Siyempre, these are some marine scientists that already there, and I'm very privileged that I am lined up with them for this webinar. With that po, maraming pong salamat at have a nice day po.
Thank you so much, Doc GB. Sir Lem, a few words from
Grabe
you.
naman. Grabe naman, pinatatanda mo naman ako, JB. Nag-teacher ko rin sila Ma'am Edna eh.
Hindi naman, sir. Grabe ka, pero syempre you're already there.
Biro lang. But anyway, ang mensahe ko simple lang, and I would rather evoke my more of being an environmental scientist. Marami tayo, iba't ibang tao ang nasa audience natin ngayon, samut-sari. I would like to make sure that this message is clear. Ang bayan po natin ay naghihirap. Ang ating environment ay nako-compromise dulot ng kahirapan. Ang palagay ko, edukasyon ay isa sa mga susi na mag-aangat ng ating mag-circumvent ng economic inequality nito. Kailangan nating ayusin, tulungan ng mga mahihirap, bigyan ng oportunidad para makapag-aral, mag-break yung circuit ng kahirapan. Pangalawa, anong magagawa natin towards that? Be passionate and be good at what you do. Kung hindi kayo scientist, that's not a problem. But for me, I would encourage that you still recognize and know some basic premise of the systematic collection on how data is provided for and how you should be able to measure what are trustworthy information, what are factual, because in the midst of all of this advancement, there's so much fake news, trolls, and the like. It's amazingly incomprehensible na napakataas na ng level ng siyensya ay napakarami pa rin hindi naniniwala. So sana gawin nating sentro ng ating polisiya, mga policies na pinidevelop, ay base sa siyensya, evidence-based. At makakatulong kayo dyan by being good at what you do at right now. Don't just say na, re-retire na rin naman ako. Be passionate at what you do and you can never go wrong with that. Maraming salamat po sa inyo lahat.
Maraming salamat din po, Doc Lem. And what a nice way to end our open forum, no? Ayan. So, I could, if meron pa po kayong mga tanong, just drop it in the chat box or you can include it later during the during or when you're doing the evaluation form. We'll have it we'll just have it together, send it to Doc JV and Doc Lem, and have a response with you as soon as possible. Okay. So before we award the certificate to our speakers, if you like our webinars and discussions, such as this one, don't forget to like and follow the socials of BRSL and NSRI. You can find us in Facebook and in YouTube. And we also share updates on our services, as well as the news and innovations in any fields of biological science. Okay, of course, our biggest thanks to our speaker, Dr. JV and Dr. Lem. And to show our appreciation, here we present these certificates of appreciation to you. And so, BRSL and NSRI in UP Diliman is awarding this certificate of appreciation to Dr. JV Sacco for having delivered a lecture during the BRSL webinar series episode 8 with the theme Karagatan para sa Kinabukasan, Exploring the Wonders of the Philippine Waters, held on the 26th day of May 2021 via Zoom. Signed by Dr. Pierre Angeli G. Vital and Dr. Elmer S. Estacio. Thank you very much, Sir JV.
Thank you po. Marami pong salamat.
Okay. Also, this is a certificate of appreciation awarded to Dr. Lemuel B. Aragones for having delivered a lecture during the BRSL webinar series with the theme Karagatan para sa Kinabukasan, Exploring the Wonders of Philippine Waters, held on the 26th day of May 2021 via Zoom. Signed by Dr. Pierre Angeli G. Vital and Dr. Elmer S. Estacio. Thank you so much, Sir Lem. And this webinar wouldn't be possible without the team behind it. Of course, job well done to the research associate of BRSL who tirelessly planned and orchestrated this webinar. And of course, to you, our audience, all thanks to you as well for taking the time to join us today. If we may invite everyone to join and open their cameras for the photo documentation. Ready na po ba ang mga ngiti natin? Ate Donna?
Hello, pwede pa stop nung share screen?
Ayan.
Okay na po.
Okay. So may 3 pages po tayo, tama po ba?
Yes po.
Okay po. I'll just count. Let me know, Ate Donna, when you're ready.
Ready na po.
Okay, so page 1, smile everyone. 1, 2, 3. Okay. Next page po. Smile pa rin tayo, ha? 1, 2, 3. Konti na lang. Last na to. Smile everyone. 1, 2, 3.
Okay na po.
Okay, thank you, Ate Donna. Of course. So, ayan. So, thank you po ulit sa mga umaten. We appreciate you being here with us. Don't forget to answer the evaluation form, especially those who will be needing an e-certificate of attendance, because only those who will complete the evaluation will get the certificate. It's posted here on the screen, also in the chat box. Just click on it and answer it. The e-certificate will be with you within the day. If you cannot answer it now, the evaluation will be available till 5 p.m. today. Ayan. So, once again, thank you everyone for coming, and we hope to see you in our next episodes. Don't forget to follow and like us for more webinars and updates, and we hope for you to stay safe. Paalam everyone. May I request our the BRSL staff and the speakers to stay stay for a photo documentation as well. Thank you.
Hello po, good morning. Thank you so much, Sir Lem and Sir JV.
Thank you
Maraming
po.
po salamat.
Thank you.
Thank you po.
Doc
Ang
Pai, thank you.
Thank you, ah.
super ganda po ng presentations niyo both.
Thanks.
Mag, pwede po bang i-request ng everyone para sa isang picture taking naman natin lahat? Ay, staff and our speakers as well. Ayan. Ganda ni Ate Jessa. Oh.
Panis. Naisingit pa yun.
And, hindi ko kita, sorry, naka-phone lang ako. Ayan.
Wala si Sir Lemuel, umalis na.
Umalis na ata si Sir Lem.
Wala.
Hindi na rin niya ata narinig. So, ayan. Sino po pong pwedeng mag-picture? Sorry, naka-phone po ako.
Go, ako na lang.
Okay. Thank you, Ate.
Okay, wait lang. Okay na. Yes. Okay, 1, 2, 3. Isa pa. Wait lang po. Wait lang po. Okay, isa pa po. Okay, 1, 2, 3. Okay na po.
Thank you, Doc Pai.
Thank you din po.
See you later sa meeting.
Meron ba?
Thank you po.
Bye. Thank you, guys.